Hello dear students, I am NEET Biology faculty working at Topaz Academy, Tirupur. We offer you this video lecture so that it would be useful for you to prepare for NEET 2020. As per government instructions, stay home, stay safe from the coronavirus infections. Today, we will start with the unit Cell Cycle and Cell Division. Cell Cycle and Cell Division unit explains you what happens during the cell growth and what happens or how a cell divides into two cells. Cell cycle explains you the cyclic changes that is happening in a cell. Cyclic changes அப்படினா என்ன அப்படினா this is going to happen regularly. அதாவது ஒரு செல் ஃபார்ம் ஆக ஆரம்பிச்சதுல இருந்து அந்த செல் ரெண்டா டிவைட் ஆக வரைக்கும் நடக்கிற cyclic changes you call it as cell cycle. இந்த செல் சைக்கிள்ல வந்துட்டு the changes that are going to happen, the first change that is going to happen is going to be cell growth. Cell growth அப்படினா நம்ம என்ன சொல்றோம் அப்படினா increase in cell size. இப்போ cell size increase ஆகிறதுக்கு cell size increase ஆகிறதுக்கு cell என்ன பண்ணும் அப்படினா it will perform all metabolic activities like taking in food, digestion of food, production of energy, and the cell-ஓட excretion. இது எல்லாமே cell-ஓட growth அப்படிங்கிறதுல வந்துட்டு this is the first thing that is happening. Second thing that is happening வந்துட்டு increase in number. Increase in number வந்துட்டு எப்படி பண்ணும் அப்படின்னா by mitotic divisions or meiotic divisions. So by these divisions cell number increase ஆக போகுது. இப்போ நம்ம cell cycle அப்படினா நமக்கு என்னன்னு தெரியணும், எங்க ஆரம்பிக்குது எங்க முடியுதுன்னு தெரியணும். We'll consider ஒரு cell, it can be any cell, ஒரு prokaryotic cell இல்லாட்டி ஒரு eukaryotic cell நம்ம கன்சிடர் பண்ணிக்கலாம். இந்த cell வந்துட்டு when it is undergoing cell cycle இது என்னென்னலாம் பண்ண போகுது, we'll consider this is a newly formed cell and it is entering into cell cycle. Any cell that is newly formed will enter into cell cycle. இப்போ இந்த cell வந்துட்டு if it is entering into cell cycle அதில் என்னென்ன changes நடக்க போகுது. Whichever cell that is entering into cell cycle is going to follow all these following events. We'll consider this is the cell entering into cell cycle. Cell cycle குள்ள enter ஆகணும் அப்படினா cell would enter into G1 phase. G1 phase is the point of entry of cell into cell cycle. இப்போ G1 phase குள்ள நுழைஞ்சதுக்கு அப்புறம் வந்துட்டு this is going to increase in size. It moves. First thing it crosses is G1 checkpoint. In later we'll see what is G1 checkpoint. After G1 checkpoint the cell is going to enter into S phase. S phase, also called a synthesis phase, is the place where the cell undergoes a DNA replication. After completion of S phase, the cell enters into G2 phase. G2 phase, it is a preparatory phase. It is going to prepare, the cell is going to prepare for M phase. That is G2 phase. The next phase is called as the M phase. M phase, it is the multiplication phase where the cell is going to divide. Multiplication can be either by mitosis or meiosis. Mitosis or meiosis happens in two stages. The first stage is called as karyokinesis or nuclear division, and then cytokinesis or the cytoplasmic division. As the end of cytokinesis, a new cell is again going to be, two new cells are again going to be formed, then again they enter back into the G1 phase. So right from G1 phase, the cell that is traveling up to G2 phase together is called as interphase. So a part of cell cycle can be called as interphase, which is subdivided into G1, S and G2 phases, and another part is called as M phase, which is divided into karyokinesis, that is nuclear division of a cell, and cytokinesis, that is the cytoplasmic division of a cell. Before we get into the details of G1, G2 and S phases, we will see what is differentiation, de-differentiation and redifferentiation processes. This mainly is concerned with cells after M phase, that is a newly formed cell. What happens to a newly formed cell after M phase? So in a cell cycle, the cell is going to enter into G phase, that is the gap phase, at G1 point. So after the end of cell division, that is after the M phase, the cell passes through the interphase. So this part is the interphase, and then it passes through M phase, comes out as a new cell. This new cell is said to be in cell cycle if it enters again into cell G1 phase. That is, if the cell is in cell cycle, the cell enters G1 phase again. The cell is said to be out of cell cycle if the cell moves to G0 phase. G0 phase indicates a quiescent phase. Quiescent phase means it is a silent phase. They are silent to cell division, but they are metabolically active cells. The cells in G0 phase, so cells in G0 phase are said to be differentiated cells. The process is called as differentiation. So differentiated cells are formed by differentiation. These differentiated cells, in case of plants, sometimes they are called as the permanent cells. So differentiation results in formation of permanent cells. These permanent cells, they are going to perform their specific functions. Few of these permanent cells, they can re-enter into cell cycle, and this is happening only for few cells. Not all cells enter back into the cell cycle. Only few cells can re-enter into cell cycle. These cells undergo a process called as de-differentiation, that is loss of differentiation process, de-differentiation, and they are entering into cell cycle. This re-differentiated cells, they can undergo again G1, S phase, G2 phase and M phase, and result in formation of new cells. These new cells again have their fate in two ways. One is they can enter into cell cycle again, or they can enter into G0 phase. So when they are entering into cell cycle, means the cells are still in cell cycle, they are able to divide. When they are entering into G0 phase, means that they are entering into the quiescent phase, that is, they are going to become silent for cell division. Now these cells that are entering into G0 phase again will be called as re-differentiated cells, and the process is called as re-differentiation, because they are getting differentiated again into permanent cells. Now we will see what is happening in the phases of the cell cycle. The first phase is interphase. In interphase, the first subdivided phase is the G1 phase. So G1 phase, we know it is the point of entry of the cell into cell cycle. And G1 phase, it is called as a preparatory phase, and the cell prepares for synthesis, for the synthesis phase, that is the S phase. So what all is going to be prepared in the cell for the S phase? The first thing, the enzymes that are required for replication are prepared, and transcription translation occurs and for this enzyme production, and hence there should be all forms of RNAs that has to be prepared by the cell. And since these are energy requiring processes, ATP synthesis also happens in the cell. And S phase involves replication of DNA, which means it requires the nucleotide formation and all substances that are required for the nucleotides, that is the nitrogen bases, pentose sugars, and the phosphoric acid group are also prepared during the G1 phase. By the end of G1 phase, the cell undergoes G1 checkpoint. G1 checkpoint means the cell is going to become committed for cell division. That is, a cell that is entering into G1 phase and completes G1 checkpoint means the cell is committed for cell division, and the cell checks for all these substances if they are ready for the S phase to occur. The second phase is S phase, which is also called as the synthesis phase. In synthesis phase, DNA replication occurs. DNA replication means formation of identical copies of the DNA that is present in the cell. So why should be these identical copies formed? Identical copies are formed because they has to be transferred to the newly formed daughter cell. That is, after M phase, newly formed daughter cells would be formed, and these daughter cells should receive the complete chromosome content. Only if they are replicated, they can receive this chromosome content. That is why the DNA replication occurs. And the next phase is going to be G2 phase. G2 phase is again a preparatory phase. For this preparation is going to be for the M phase. So in M phase, the substances that are to be prepared are, first thing, RNA. This is for transcription and translation for formation of proteins. So what are the proteins that are required for M phase to occur? The proteins that are mainly required are the tubulin proteins for formation of aster rays, spindle fibers, etc. and the plasma membrane proteins. Because cell division is going to occur, a new plasma membrane has to be formed. So plasma membrane proteins are also to be ready, kept ready by the cell. So these are substances that are formed. And ATP, it is an energy source which is going to be used by the cell for cell division process. We will also have to study what is a chromosome value. Chromosome value can be also called as the C value of a cell. C value of a cell means the amount of chromosomes present in the cell, the nucleus of the cell. And before that, we have to know the DNA content. When we call a DNA content of the cell, DNA content denotes the number of sets of chromosome present in a cell. So sets of chromosome we call it as ploidy, which can be given by the letter N. And the number of sets of chromosome normally we talk about is a diploid set. Let us consider a diploid set of chromosomes. In a diploid cell, the DNA content is equal to 2n, and the chromosome value or the C value of a cell is equal to 2C, means 2 copies of a single chromosome is present. This diploid cell enters into G1 phase. So in G1 phase, the DNA content of a cell is going to be 2n, and the C value is also going to be 2C. Now the cell enters into S phase. In S phase, replication happens, means the number of cells, that is the chromosome value, is going to be doubled because of replication by formation of identical copies. But the DNA content remains the same because they are still a diploid cell, whereas the C value of a cell will be called as 4C because the 2C has multiplied by 2 by a replication process. So this undergoes replication and it has been multiplied, forms a 4C chromosome value. Now this one enters into G2 phase. G2 phase also the DNA content is going to be 2n and the C value is going to remain the same as 4C. The cell is going to now enter into M phase. The first phase is karyokinesis. In karyokinesis, the cell nucleus alone divides. This results in formation of 2 nuclei, and the DNA content of the cell is going to be still same, the 2n, and the C value also remains as 4C because they are present within the same cell. After cytokinesis process, there are 2 cells that are formed now, and the DNA content is going to be the same 2n because it is the diploid cell that is dividing, but the chromosome value again reduces into 2n because the 2 cells are divided into 2 new cells. Because of this, the chromosome value again reaches 2n. Thus, the cell division, it starts with a 2n cell with a chromosome value of 2C and ends in formation of a 2n cell with a chromosome value of 2C. Now, we will start with the cell division processes or cell division types. There are 3 different types of cell division that normally occurs. First type is called as amitosis, the second type is called as mitosis, and third type is called as meiosis. Amitosis is also called as a direct division of cell. This is called as direct division because karyokinesis and cytokinesis happens simultaneously, and cytokinesis both are going to happen simultaneously, and the cell divides approximately into 2 equal halves. And the division of the nucleus as well as the cell divides by invagination of their membranes. So a cell, will consider the cell with its nucleus, and if it goes on with a mitotic division, the cell first starts its invagination with the nuclear membrane. Meantime, the cytoplasmic membrane, that is the plasma membrane of the cell, also starts invaginating, and finally they pinch off into two equal sized cells. This is approximate division. Two equal sized cells with their own nucleus. Example of cells taking up a mitosis is a few bacteria, cyanobacteria, and in case of plant cells, endosperm division can be by amitosis, and in case of animal cells, so this is in case of plants, and in case of animal cells, cartilage cells divide by amitosis. But the disadvantage of this amitotic division is there is no surety of equal distribution of the chromosomes, because this is an approximate dividing cell into two halves. So there is no surety for equal distribution of the chromosomes of the cell. The next type of cell division, mitosis. Mitosis is also called as indirect division or equational division. This is because their chromosome number gets equally divided into two halves into the two daughter cells that are formed. The mitosis was first reported in plant cells by Strasburger, and in case of animal cells by Fleming, and he is the person who coined the term mitosis also, Fleming. And this division happens in, so where does mitotic division happen? This happens in vegetative cells. Vegetative cells can be also called as the somatic cells, or it can be also said as the non-reproductive cells. They can also happen, so this is one cell in which it is happening. They can also happen in germ cells also for increase in number of the germ cells. The next is meiotic division. Meiotic division, it is also called as reductional division because the number of chromosomes that is received by the daughter cells is going to be reduced than the parent cell. And this was first reported, meiotic division was first reported in the egg cells of sea urchin. It is a type of echinoderm. It was defined in these cells, and it was given by a person called Van Wende. And this meiosis, the cells that is undergoing meiosis are called as meiocytes, and meiocytes are usually only reproductive cells, or which can be also called as the germ cells. Mitotic division can happen in both diploid and haploid cells, whereas meiotic division happens only in diploid cells. We will now start with the mitotic divisions. So mitosis can happen both in diploid cells as well as haploid cells. In a diploid cell, when mitosis occurs, this diploid cells results in formation of two diploid cells, and if it occurs in a haploid cell, this results in formation of two haploid cells. This is a reason why this mitotic division is also called as the equational division. And the steps involved in mitotic division. Any cell that has to undergo mitotic division has to first cross the interphase. So the interphase, it goes on into the G1 phase, prepares itself for S phase. So this is going to be a preparatory phase. And it undergoes S phase, during which a DNA replication occurs, as well as in case of animal cells, centriole division also occurs. So centriole division is more specific only in case of animal cells. This is because plant cells do not have centrioles. And the next phase is going to be G2 phase, which is going to be again a preparatory phase for M phase. And this is the first step in mitotic division. And coming to the next step, it is going to be the M phase. So M phase is again divided into two parts. The first one is karyokinesis, and the second step is cytokinesis. Karyokinesis in mitosis is again divided into 4 subphases. The first phase is called as prophase. The second phase is called as metaphase. The third phase is called as anaphase. And the last subphase is called as telophase. By the end of telophase, this nucleus would have been divided into 2 equal halves. And cytokinesis is going to be cytoplasmic division. Cytoplasmic division is going to be different for plant and animal cells. This is because of the presence of the cell wall and absence of cell wall in animal cells. In case of plant cells, the cytokinesis is by cell plate method. And in case of animal cells, the cytokinesis is by invagination of plasma membrane. So to start with, the karyokinesis process, the first phase is going to be prophase. In prophase, the cell is going to prepare itself for karyokinesis, that is nuclear division. So karyokinesis is nothing but nuclear division, and the cell is going to prepare itself for nuclear division. இப்போ ப்ரோஃபேஸில் செல் என்ன பண்ணும் அப்படின்னா, it is going to prepare itself. இப்போ ப்ரிப்ரேஷனுக்கு என்ன பண்ணும் first? If this is the, we'll consider a cell with its nucleus and their chromatin fibers, that is their genetic material. So this is going to be the plasma membrane. This is the nuclear membrane, and these are the chromatin fibers. So cell during prophase, what happens is, there are many happenings that is going to occur during prophase. First வந்து என்ன அப்படின்னா, first வந்துட்டு nuclear membrane disintegrate ஆகும். அதாவது it would be lost. So nuclear membrane disintegrate ஆகும், and this structure usually we call it as the nucleolus. So second function is nucleolus disintegration. So nucleolus disintegrate ஆகும், அதாவது உடைஞ்சு காணாமல் போகிறது தான் disintegration. Third, organelles. All organelles except centrioles. ஏன்னா சென்ட்ரியோல்ஸ் டிவைட் ஆகிருக்கு, சென்ட்ரியோல் கண்டிப்பாக செல்லில் இருக்கணும் செல் டிவிஷனுக்கு. ஸோ சென்ட்ரியோல்ஸ் டிஸ்இன்டகிரேட் ஆகாது, centrioles will be present and other organelles disintegration நடக்கும். அடுத்தது chromatin fibers. இந்த chromatin fibers வந்துட்டு they'll start condensing. So condensation of chromatin fiber start ஆகும். So condensation வந்துட்டு result in formation of chromosome structure. The next thing that is happening is centrioles. So centrioles already S phase la divide ஆகிருந்த centrioles வந்துட்டு they'll start moving towards the pole. So poles அப்படின்னா usually the ends of the cell. அதை தான் போல்ஸ்னு சொல்லுவோம். The centrioles are going to move to the poles. இது ஏன்னா centrioles already S phase la divide ஆகிடுச்சு, they'll move to the poles. Then formation of aster rays. Aster rays அப்படின்னா these are short microtubular structures around centrioles. So centrioles சுற்றியும் short microtubular structures form ஆகும், அதுக்கு பேர் aster rays. Then formation of spindle fibers. Spindle fibers அப்படின்னா these are long microtubular structures. இந்த microtubular structures வந்துட்டு they are going to extend from one pole to another pole. So ஒரு centriole பக்கத்தில் ஆரம்பிச்சு it moves to another pole. இதை வந்துட்டு நம்ம spindle fibersனு சொல்லுவோம். இந்த spindle fibersல வந்துட்டு there are two types. First one, it is called as continuous spindle fiber. Continuous spindle fiber எந்த ஒரு டிஸ்டர்பன்ஸும் இல்லாமல் ஒரு எண்டிலிருந்து இன்னொரு எண்டில் வரைக்கும் இருக்கும். So it is also called as interpolar fibers. இன்னொன்று வந்துட்டு discontinuous spindle fiber. எதனால் discontinuous அப்படின்னா on their way they will be attached to the chromosomes. So chromosome கூட attach ஆகிருக்கிறதுனால they will be also called as chromosomal spindle fiber. So இது எல்லாமே வந்துட்டு ப்ரோஃபேஸில் நடக்க போகுது. So when we show it in a cell, so இந்த செல்லில் வந்துட்டு it is will show it clearly. Centrioles will be present at the poles and அதை சுற்றியும் short fibers, these are called as aster rays. So this centrioles plus aster rays is together called as aster. So இங்கே வந்துட்டு இந்த ஸ்ட்ரக்சர் வந்துட்டு we call it as an aster. This contains centrioles and the aster rays. அதுக்கப்புறம் spindle fibers. ஸோ ஒரு எண்டிலிருந்து இன்னொரு எண்டு வரைக்கும், ஒரு போலில் இருந்து இன்னொரு போல் வரைக்கும் போகிறது வந்துட்டு continuous spindle fibers. And these are continuous spindle fibers that are formed. In between la vanditte if they are attached to chromosomes. So chromosome structure condensation la form aayirukkum by condensation of the chromatin fiber to chromosome structure. In between la if the spindle fibers are attached to these chromosomes, they will be called as the discontinuous spindle fiber. இது வந்துட்டு discontinuous spindle fiber that is formed, and the aster plus spindle fiber together will be called as the spindle apparatus. Now cell is entering into metaphase. இப்போ மெட்டாஃபேஸில் வந்துட்டு என்னென்ன நடக்கும் அப்படின்னா in metaphase இந்த செல்லில் இருக்கிற அந்த குரோமோசோம்ஸ் they will move to the metaphase plane. So these are the centrioles, asters, and the continuous spindle fibers and metaphase நடக்க போகிற ஈவெண்ட் என்ன அப்படின்னா the chromosomes are arranged in the metaphase plane. Metaphase plane also called as equatorial plane. இந்த பிளேனில் அரேஞ்ச் ஆகும். ஸோ ஈக்குவட்டோரியல் பிளேன் அப்படிங்கிறது வந்துட்டு more or less it is going to be the center of the cell. இன்னொரு ஈவெண்ட் என்ன அப்படின்னா the chromosomes will have the spindle fibers attached on both their sides. So spindle fibers chromosomes-ஓட ரெண்டு சைடும் அட்டாச் ஆகிருக்கும். ரெண்டு சைடு அட்டாச்மெண்ட் அப்படின்னா the spindle fiber is going to come from opposite poles. So ஒரு side attachment is with one spindle fiber from one pole and இன்னொரு side attachment is with spindle fiber coming from another pole. And by the end of metaphase the cell undergoes metaphase checkpoint. So metaphase checkpoint அப்படின்னா என்ன அப்படின்னா cell is going to check for these two conditions. அதாவது chromosomes வந்துட்டு ஈக்குவட்டோரியல் பிளேனில் அரேஞ்ச் ஆகிருக்கா அப்படின்னு செக் பண்ணுறது. இன்னொன்று வந்துட்டு if the chromosomes are attached to spindle fibers on both the sides, ஆனால் both the sides attachment வந்துட்டு spindle fibers has to come from opposite poles. இதை வந்துட்டு செக் பண்ணுறது தான் வந்துட்டு metaphase checkpoint. ஸோ எப்படி attachment இருக்கும் அப்படின்னா usually chromosomes-ல வந்துட்டு they will be attached to the centromere region. So if this is the chromosome structure, this part is the centromere region. So centromere-ல வந்துட்டு வெளியில் கைனட்டோகோர் அப்படின்னு ஒரு protein first deposit ஆகும். So this is a கைனட்டோகோர். Kinetochore will get deposited on. So this is actually a protein on centromere region. இந்த கைனட்டோகோர் கூட ஸ்பிண்டில் ஃபைபர்ஸ் would attach. இப்போ ஸ்பிண்டில் ஃபைபர்ஸ் எங்கேருந்து வரணும் அப்படின்னா they have to come from opposite poles. That is attachment to the centromere region of the chromosome structure. இது தான் வந்துட்டு மெட்டாஃபேஸ் செக் பாயிண்டில் செல் வந்துட்டு செக் பண்ணும். ஸோ வாட் ஃபார் இந்த செக்கிங் பண்ணுறது அப்படின்னா திஸ் இஸ் ஃபார் ஈக்குவல் division of chromosomes to daughter cells. ஸோ இந்த டிவிஷனை ஈக்குவேஷனல் டிவிஷனை என்ஷூர் பண்ணுறதுக்காக தான் வந்துட்டு குரோமோசோம்ஸ் வந்துட்டு அட்டாச் ஆகிருக்கான்னு செக் பண்ணுறது மெட்டாஃபேஸ் பிளேனில். The next phase is going to be anaphase. Anaphase-ல வந்துட்டு the cell is going to separate the chromosomes to both the poles. So anaphase-ல என்னென்ன நடக்க போகுது அப்படின்னா the chromosomes that are attached to the spindle fibers are pulled towards the poles. So இந்த pulling எப்படி நடக்கும் அப்படின்னா by contraction of the spindle fibers. So spindle fibers they'll start contracting, pull the chromosomes to the opposite poles. So நான் இங்கே ரெண்டு chromosome காமிச்சிருக்கிறேன். I'll show you with one alone so that it is easy to understand. So ஒரு chromosome இப்போ மெட்டாஃபேஸ் பிளேனில் இருக்குது, ஈக்குவட்டோரியல் பிளேனில் இருக்குது. இது கூட ரெண்டு சைடும் spindle fiber attach ஆகிருக்கு. இந்த spindle fiber this is coming from this pole and this spindle fiber is coming from this pole. So this spindle fiber starts contracting in this direction, pull it towards the pole, and this spindle fiber contracts in this direction, pull it towards this pole. So that is what is pulling of chromosomes towards pole by contraction of spindle fibers. இது starting of anaphase la. Late anaphase la, that is by the end of anaphase, chromosomes reach their poles. So chromosomes are getting separated and reach the poles. So now நமக்கு தெரியும் chromosomes ஓட centromere regionல, so if this is the chromosome, to the kinetochore spindle fibers ரெண்டு போல்ஸில் இருந்தும் அட்டாச் ஆகிட்டு இது இந்த போலுக்கும் இது இந்த போலுக்கும் புல் பண்ண போகுது. When it is pulling like this, the chromosomes are going to get separated. செப்பரேட் ஆகுது அப்படின்னா இட் இஸ் யூஷுவலி இந்த ரெப்ளிகேட்டட் காப்பி இருக்கு இல்லையா, அந்த ரெப்ளிகேட்டட் காப்பி செப்பரேட் ஆகும் இந்த மாதிரி. இப்படி செப்பரேட் ஆகும்போது வென்எவர், ஸோ இந்த ரெப்ளிகேட்டட் காப்பியை வந்து நம்ம சிஸ்டர் குரோமேட்டிட்ஸ் அப்படின்னு சொல்லுவோம். இந்த சிஸ்டர் குரோமேட்டிட்ஸ் இப்படி செப்பரேட் ஆகி இது இந்த போலுக்கும் இது இன்னொரு போலுக்கும் போகும்போது இந்த சென்ட்ரோமியருக்கு நடுவில் தேர் வுட் பி அனதர் டைப் ஆஃப் ஸ்பிண்டில் ஃபைபர் ஃபார்ம்டு. So if this is one moving to one pole and if this is another sister chromatid moving to another pole, நடுவில் வந்துட்டு there is another type of spindle fiber that is formed. This type of spindle fiber is called as interzonal fiber. இப்போ interzonal fiber ஃபார்ம் ஆகும். இந்த இன்டர்சோனல் ஃபைபர் எதுக்காக ஃபார்ம் ஆகுதுன்னா, இங்கே ஒரே ஒரு காமன் குரோமோசோமாக இருந்தது, இங்கே ரெண்டு சென்ட்ரோமியராக பிரிஞ்சிருக்கு. இந்த ரெண்டு சென்ட்ரோமியராக பிரிக்கும்போது there is splitting of centromere. ஸோ இந்த ஸ்ப்ளிட்டிங் நடக்கும்போது அதோட ஸ்டெபிலிட்டியை மெயின்டைன் பண்ணுறதுக்காக நடுவில் ஒரு இன்டர்சோனல் ஃபைபர் ஃபார்ம் ஆகுது. திஸ் ஒன் இஸ் த தேர்ட் டைப் ஆஃப் ஸ்பிண்டில் ஃபைபர். இது இன்டர்சோனல் ஸ்பிண்டில் ஃபைபர். ஸோ அனாஃபேஸோட எண்டில் chromosomes வந்துட்டு they would have reached the poles. The next step, telophase, அதாவது the end of the last phase of the karyokinesis process. இப்போ karyokinesis ஓட end அப்படின்னாலே வந்துட்டு நியூக்ளியர் டிவிஷன் கம்ப்ளீட் ஆகிருக்கணும். ஸோ telophase வந்துட்டு it can be distinguished as it is the reverse of ப்ரோஃபேஸ். அதாவது ப்ரோஃபேஸில் புதுசாக ஃபார்ம் ஆன ஸ்ட்ரக்சர்ஸ் இங்கே டிஸ்இன்டெக்ரேட் ஆகும், டிஸ்இன்டெக்ரேட் ஆன ஸ்ட்ரக்சர்ஸ் புதுசாக ஃபார்ம் ஆகும். ஸோ டிஸ்இன்டெக்ரேட்டட் ஸ்ட்ரக்சர்ஸ் இன் ப்ரோஃபேஸ் வந்துட்டு they are going to be nuclear membrane, nucleolus, organelles. இதெல்லாம் டிஸ்இன்டெக்ரேட் ஆச்சு. ஸோ these are going to be now newly formed in both the poles. அடுத்தது நியூலி ஃபார்ம்டு ஸ்ட்ரக்சர்ஸ் வந்துட்டு, அதாவது ப்ரோஃபேஸில் நியூலி ஃபார்ம்டு ஸ்ட்ரக்சர்ஸ் என்னென்ன அப்படின்னா ஸ்பிண்டில் அப்பேரட்டஸ், அதாவது ஆஸ்டர் ரேஸ், சென்ட்ரியோல்ஸ் அண்ட் த ஸ்பிண்டில் ஃபைபர்ஸ். ஸோ இதுதான் வந்துட்டு நியூலி ஃபார்ம்டு ஸ்ட்ரக்சர்ஸ். இந்த ஸ்ட்ரக்சர்ஸ் வந்துட்டு டிஸ்இன்டெக்ரேட் ஆகும். ஸோ இதெல்லாம் டிஸ்இன்டெக்ரேட் ஆகிட்டு இந்த ஸ்ட்ரக்சர்ஸ் புதுசாக ஃபார்ம் ஆகும்போது ரெண்டு போல்ஸில் ரெண்டு புது நியூக்ளியஸ் ஃபார்ம் ஆக போகுது. இன்னொரு பாயிண்ட் we have to remember is the chromosome structure. Condensationல ஃபார்ம் ஆன chromosome structure is going to be lost and results in formation of chromatin fiber. So DNA திரும்பவும் வந்துட்டு it is going to become like chromatin fiber. So two nucleus has been formed as a result of the last step of the karyokinesis, that is telophase. Now the cell enters into cytokinesis. Cytokinesis we have already seen it is different for plant cells and animal cells. In case of animal cells, cytokinesis is a simple process. It is going to be by the invagination of the plasma membrane at the equatorial plane. So it is going to be invagination of the nuclear membrane resulting in formation of two new cells. So in the invagination வந்துட்டு it is going to grow, result in forming two cells. So in the invagination it is finally it will fuse to form two cells. As a result of cytokinesis in animal cells by invagination, 2 new cells are being formed. In the cells, again they are going to grow into their original size, enter into G1 phase or G0 phase depending on the type of cell that is involved. In case of cytokinesis in plant cells, because plant cells la cell wall irukra reason nala cytokinesis vanditu is not going to be by invagination, because the cell wall is going to be a rigid membrane. Adanala vanditu plant cells take up a method called as cell plate method. So what is this cell plate method? For this, cells enna pannum appadina, first step vanditu formation of phragmoplast. So இந்த phragmoplast அப்படின்னா என்னன்னா, these are the disintegrated Golgi and ER. In the structures, they are going to first deposit at the equatorial plane. இந்த ஸ்ட்ரக்சருக்கு பேர் வந்துட்டு phragmoplast. After that, phragmoplast, next is formation and growth of cell plate. So இந்த phragmoplast settle ஆயிருக்கு இல்லையா, இந்த phragmoplast they will start secreting and deposit calcium pectate. So once they secrete calcium and they deposit this calcium pectate on the phragmoplast, they will be called as the cell plate. In the cell plate vanditu it grows to the entire length of the cell. The last step is formation of new cell wall. So once cell plate, that is calcium pectate deposition, cell plate form ஆன உடனே the 2 new cells starts depositing the primary cell wall on the cell plate. So thus new cell wall is formed. So to represent, we'll show a plant cell in this way itself, because this is the most common way of drawing a plant cell. In the plant cell, so this part is going to be the plasma membrane part that was initially formed, and let us consider these are the 2 nuclei that are newly formed. இங்க இருக்கு ரெண்டு போல்ஸ்லயும். இப்போ ஃபர்ஸ்ட் ஸ்டெப் வந்துட்டு phragmoplast deposition. ஸோ இந்த டிஸ்இன்டகிரேட் ஆன ப்ரோஃபேஸில் டிஸ்இன்டகிரேட் ஆன எண்டோபிளாஸ்மிக் ரெட்டிகுலம் பார்ட்ஸும் கோல்கியோட பார்ட்ஸும் வந்துட்டு ஃபஸ்ட்டு டெபாசிட் ஆகிறது. திஸ் ஒன் யூ கால் இட் ஆஸ் த phragmoplast. இப்போ இந்த phragmoplast வந்துட்டு அடுத்தது வந்துட்டு it is going to grow in size as well as it is going to deposit calcium pectate. So இப்போ calcium pectate வந்துட்டு, so it is going to completely cover the entire length, அந்த equatorial plane ஓட entire length cover பண்ணிட்டு they would start depositing calcium pectate. இப்போ calcium pectate deposit பண்ணின உடனேயே வந்துட்டு செல், இந்த ரெண்டு செல்லும், அதாவது ஒரு சைடும் இன்னொரு சைடும், இது வந்துட்டு it will act as 2 cells and they start depositing. So this part you call it as cell plate. இப்போ இந்த cell plate வந்துட்டு once it is formed, cell plate form ஆன உடனே cell வந்துட்டு considers that cell plate as a middle lamella. ஸோ இதுதான் பின்னாடி வந்துட்டு ஒரு செல்லுக்கும் இன்னொரு செல்லுக்கும் நடுவில் மிடில் லேமல்லாவை ஆக்ட் பண்ண போகுது. The cell is going to form the new cell wall. The primary cell wall வந்துட்டு form பண்ணிக்கும். So primary cell wall and the complete plasma membrane structure with its new nucleus. So this is cytokinesis in a plant cell by cell plate method. So this is the end of mitosis. So these are two new cells formed. The next type of cell division is meiosis, also called as the reductional division. இது எதனால reductional division அப்படின்னா, meiosis can happen only in diploid cells. In the diploid cells would undergo meiosis 1, அதாவது meiosis-ல ரெண்டு ஸ்டெப் இருக்குது, meiosis 1 and meiosis 2. இதில் it undergoes meiosis 1 to form 2 haploid cells. இந்த ரெண்டு haploid cells-உம், so these are haploid cells that are formed as a result of meiosis 1, and this haploid cells, both the haploid cells are going to undergo meiosis 2. So meiosis 2 form பண்ணும்போது what is obtained is 4 haploid cells. So these are all haploid cells. So because it is called reductional division, because a diploid cell undergoes meiosis, results in formation of a haploid cell. So the chromosome set is reduced by half. So it is called as a reductional division. And the steps involved in meiosis process. So meiosis itself is divided into two major phases. One is called as meiosis 1, the other one is called as meiosis 2. The meiosis 1 is again divided into two subphases. The first one you call it as karyokinesis 1, and the next phase is cytokinesis 1. And meiosis 2 again is divided into two phases, that is karyokinesis 2 and cytokinesis 2. The karyokinesis 1 is again subdivided into 4 phases. The first one you call it as prophase 1, metaphase 1, anaphase 1 and telophase 1. This prophase 1 is again divided into 5 subphases. They are leptotene, zygotene, tachytene, diplotene and diakinesis. So all these 5 phases or subphases of prophase 1 continues with metaphase 1, anaphase 1, telophase 1 and cytokinesis 1. And after completion of meiosis 1, so after meiosis is completed, the cell undergoes a short preparatory phase for meiosis 2 called as interkinesis. This is purely a preparatory phase without DNA replication. So there is no DNA replication before meiosis 2. This is just preparation for meiosis 2. And after interkinesis, the cell enters into both the cells that are formed from cytokinesis 1 enters into meiosis 2. In meiosis 2, the cell undergoes karyokinesis 2 and cytokinesis 2. Karyokinesis 2 is again divided into 4 subphases, that is prophase 2, metaphase 2, anaphase 2 and telophase 2. And then from telophase 2 the cell enters into cytokinesis 2, results in formation of 2 cells from each cell from meiosis 1. Thus here it is going to be a diploid cell, one cell, and after meiosis there are going to be 2 haploid cells formed, and after cytokinesis 2 there are going to be 4 haploid cells that are formed. So for any cell entering into the meiosis process, so if a cell is entering into meiosis, the cell undergoes the interphase, that is the G1 S phase and G2 phase, and after that it enters into the M phase. So it is a replicated cell that is entering into M phase. So in M phase it starts with meiosis 1 karyokinesis. Karyokinesis going to start with the prophase 1. In prophase 1 the first stage is going to be, the substage is going to be the leptotene stage, or also called as the Bonket stage. And the events that happens during the leptotene stage I have written it here, and to explain it with a representation, so the nuclear membrane, nucleolus are all going to remain intact, and the chromatin fibers have just started condensing, and these condensing chromatin fibers, they remain attached to the nuclear membrane at one end. So one of their end is going to be attached to the nuclear membrane and the other end remains free, and this remains of a structure of a bouquet, so called as the Bonket stage. The next stage is the zygotene stage. So in zygotene stage, so this is leptotene stage. The next stage is going to be the zygotene stage, and in zygotene stage the differences that is going to occur within the nucleus is, I'm going to represent it with a single set of homologous chromosomes. So when we say homologous chromosomes, they are always going to occur in pair. We'll consider this is a homologous pair which is been formed by condensation of this chromatin fiber. So 2 of these, let us consider, I am just considering this one, and these 2 chromatin fibers have condensed into these structures. These 2 are the homologous pairs. So these homologous pairs of chromosomes, now they are going to start the crossing over process by synaptonemal complex and chiasmata formation. But to understand this synaptonemal complex and chiasmata formation, we have to know what is a dyad and a tetrad. If it is a homologous pair, we know that these homologous pair have already replicated in the S phase. So each of these chromosome has to look like this, that is they have their replicated sister copy with them. So this one, for a homologous pair, it is called as, this structure is called as a dyad, and this dyad has their sister chromosomes that is very closely attached to each other, such that it looks like a single chromosome alone. So they are very closely attached, so it looks like a single chromosome. This structure we call it as a dyad. But to undergo synaptonemal complex and chiasmata formation, the sister chromatids, the chromatids alone gets splitting. So there is splitting of chromatids. So chromatids split, resulting in formation of this structure, that is each chromosome having 4 arms. This structure you call it as a tetrad. So now this tetrad is undergoing chiasmata formation. Chiasmata formation abdina, it is an X-shaped cross formed. Any X-shaped cross will be called as chiasmata. இதில் the X-shaped cross வந்துட்டு will be formed within tetrads. இந்த homologous chromosome these two arms, this arm and this arm together, we call it as sister chromatids. Whereas this arm, that is the arm of one homologous chromosome and its pair, these two, we call it as non-sister chromatids. So there is going to be crossover between the non-sister chromatids. So non-sister chromatids undergo the crossing over process. So what is this crossing over process is, these two chromatids, they come closer to each other and they are going to remain like this. So there can be as many as crossovers that can be formed in a chromosome, but to make it simpler and easier, we are representing only a single crossover. This structure that is formed is called as chiasmata, and it remains like this, that is in this crossed over position with the help of synaptonemal complex. So synaptonemal complex அப்படினா ஒன்றும் இல்லை, இது வெறும் group of proteins that is going to maintain the chiasmata structure. This structure formation happens in zygote stage. அடுத்தது வந்துட்டு packetine stage. packetine stageல வந்துட்டு actual crossover occurs, that is recombinase enzyme வந்துட்டு helps in recombination process. அதாவது we'll make it look different, that is we'll shade out one chromosome so that we can differentiate between the homologous pairs. So actual crossover அப்படினா என்னன்னா, that is the exchange of genetic material between the non-sister chromatids. So crossoverல வந்துட்டு they are going to remain like this. So when we say actual crossover, என்ன நடக்கும் அப்படின்னா, இந்த recombinase enzyme வந்துட்டு they would exchange these parts of the chromosome. இந்த exchanged part இருக்கு இல்லையா, இந்த பார்ட்டை வந்துட்டு they are going to cut it and put it in the other chromosome. So this is one chromosome of the homologous pair, and this part of the chromosome, இந்த part வந்துட்டு it would come to the other chromosome. So this one gets it part up, this chromosome gets a part of this one. So this process you call it as crossover or recombination, and this helps this occurs with the help of recombinase enzyme. So this is what is going to happen for all pairs of chromosomes that are going to be present in the nucleus, but we have represented only for a single set of chromosomes so that it is easy to understand. After the actual crossover process, that is exchange of genetic material, செல் வந்துட்டு டிப்ளோட்டின் ஸ்டேஜ்க்கு போகும். இங்கே டிப்ளோட்டின் ஸ்டேஜில் இந்த கிராஸ் ஓவர் முடிஞ்ச குரோமோசோம்ஸ், அதாவது இந்த நான் சிஸ்டர் குரோமேட்டிட்ஸ் வந்துட்டு they are going to get separated by terminalization process. ஸோ இப்போது நான் சிஸ்டர் குரோமேட்டிட்ஸ் அவங்க கிராஸ் ஓவரில் வந்துட்டு they are going to be present like this. So they have received the part of their non-sister chromosome ஒரு பார்ட் வந்துட்டு அவங்களுக்கு கிராஸ் ஓவரில் கிடச்சிருக்கு. இது வந்துட்டு it is going to get separated. So separationல வந்துட்டு they will get separated into the actual tetrad form. இந்த tetrad formல வந்துட்டு the crossover part is also taken out. இந்த ப்ராசஸ் வந்துட்டு is called as terminalization. எதனால் terminalization அப்படின்னா, the two chromosomes terminalization எப்படி நடக்கும்னா, if these are the crossover regions, they are going to get separated, they move like this, get separated at their ends. ஸோ இந்த மாதிரி மூவ் ஆகிட்டு எண்ட்ஸில் இருந்து செப்பரேட் ஆகிறதுனால they are called as terminalization process, and this is also called as a zipper fashion. So this happens in diplotin stage. So by the end of diplotin stage, cell வந்துட்டு the chromosomes would have been exchanged their genetic material between the homologous chromosomes. இந்த நெக்ஸ்ட் ஸ்டெப் வந்துட்டு டயாகைனசிஸ். டயாகைனசிஸில் வந்துட்டு மைட்டோசிஸில் ப்ரோஃபேஸில் என்னென்னலாம் நடக்கும்னு பார்த்தோமோ அது எல்லாமே வந்துட்டு மியாசிஸில் ப்ரோஃபேஸ் 1ஓட டயாகைனசிஸ் பார்ட்டில் நடக்க போகுது. தட் இஸ் நியூக்ளியர் மெம்பரேன், நியூக்ளியோலஸ், ஆர்கனல்ஸ் டிஸ்இன்டெகிரேட் ஆகும். சென்ட்ரியோல்ஸ் they are going to move towards the poles and there is spindle apparatus formation. So aster rays formation, continuous spindle fiber formation and discontinuous spindle fibers. So discontinuous spindle fiber with chromosomes and continuous spindle fiber moving from one end to the other end. இந்த மாதிரி வந்துட்டு டயாகைனசிஸில் ஃபார்ம் ஆகும். And next stage is metaphase. Metaphase வந்துட்டு first they are getting arranged in the equatorial plane. So this is the equatorial plane. They are going to get arranged in the equatorial plane and they are attached to spindle fiber coming from only one pole. ஸோ ஹோமோலோகஸ் பேர்ஸ் வந்துட்டு எப்படி அரேஞ்ச் ஆகிருக்கும் அப்படின்னா ஸ்பிண்டில் ஃபைபர் ஒரு எண்டில் இருந்து வர்றது மட்டும்தான் வந்துட்டு ஹோமோலோகஸ் பேர்ஸ் கூட அட்டாச் ஆகிருக்கும். ஸோ இஃப் திஸ் இஸ் ஒன் ஹோமோலோகஸ் பேர், ஒரு குரோமோசோம் வந்துட்டு இட் வில் பி அட்டாச் ஃப்ரம் ஸ்பிண்டில் ஃபைபர் கம்மிங் ஃப்ரம் ஒன் போல். அதோட இன்னொரு பேர் வந்துட்டு வில் பி அட்டாச் டு ஸ்பிண்டில் ஃபைபர் கம்மிங் ஃப்ரம் அனதர் போல். திஸ் இஸ் வாட் இஸ் செக்ட் இன் மெட்டாஃபேஸ் செக் பாயிண்ட். நவ் அனாஃபேஸ் 1ல் வந்துட்டு ஸ்பிண்டில் ஃபைபர் கான்ட்ராக்ஷன் நடக்கும். ஸோ ஸ்பிண்டில் ஃபைபர் கான்ட்ராக்ஷனில் வந்துட்டு they are going to pull the chromosome attached to them. இப்போது இங்கே என்ன ஆகும் அப்படின்னா because homologous pairs they are attached to different spindle fibers. ஸோ ஒரு homologous pairல வந்துட்டு if it is attached to different spindle fibers we'll consider like this. அதாவது ஒரு டெட்ராடை வந்துட்டு நான் ஒரு டயாடு மாதிரி காமிக்கிறேன். ஸோ this is going to be like this. So we'll consider another pair of chromosome. So இதுவும் டெட்ராட் வந்துட்டு நான் டயாடு மாதிரி காமிக்கிறேன். So this is one pair of chromosome, this is another pair of chromosome. ஒவ்வொரு பேர்லேயும் வந்துட்டு they are going to be attached only to the opposite poles. ஸோ இந்த போலில் அட்டாச் ஆகிருக்கிறது இந்த சைட் புல் பண்ண போகுது, இந்த போலில் அட்டாச் ஆகிருக்கிறது இந்த சைட் புல் பண்ண போகுது. ஸோ பை த எண்ட் ஆஃப் அனாஃபேஸ் வாட் வுட் ஹேப்பன் இஸ் in a cell the homologous pairs. ஸோ இது ரெண்டும் ஒரு பேர், இது ரெண்டும் இன்னொரு பேர். இது என்ன ஆகும் அப்படின்னா இந்த பேர் வந்துட்டு ஒரு எண்ட் ஆஃப் த போலுக்கும், அதாவது ஒரு எண்ட் ஆஃப் த செல்லுக்கும், இன்னொரு பேர் வந்துட்டு இன்னொரு போலுக்கும் வந்துட்டு they are going to move. ஸோ இப்போ என்ன ஆகும் அப்படின்னா homologous pairs வந்துட்டு they are getting separated. If homologous pairs are separated to n condition, so இந்த அனாஃபேஸில் வந்துட்டு what happens is 2n conditionல இருந்த செல் வந்துட்டு it is converted into n condition. அதாவது n condition நியூக்ளியஸாக வந்துட்டு they are getting separated and because of this reason, ஸோ இங்கே வந்துட்டு சென்ட்ரோமியர் ஸ்பிளிட்டிங் நடக்காத ரீசன்னால் இன்டர்சோனல் ஃபைபர் ஃபார்மேஷன் கிடையாது. ஸோ தேர் இஸ் நோ இன்டர்சோனல் ஃபைபர் ஃபார்மேஷன் பிகாஸ் சென்ட்ரோமியர் ஸ்பிளிட் ஆகலை, குரோமோசோம்ஸ் தான் வந்துட்டு ஸ்பிளிட்டிங் ஆகிருக்கு. ஸோ வாட் இஸ் கெட்டிங் செப்பரேட்டட் ஹியர் இஸ் த ஹோமோலோகஸ் குரோமோசோம்ஸ் ஆர் கெட்டிங் separated. So the next is telophase 1. So telophase 1 is going to be the reverse of diakinesis. So reverse of diakinesis அப்படினா, so if this is the cell after anaphase, so diakinesis oda reverse அப்படினா வந்துட்டு formation of nuclear membrane on both the poles and the chromosomes are going to decondense to form chromatin fibers and there is organelle formation. So there is organelles that are formed and nucleolus is also formed and the spindle apparatus is going to disintegrate. So the telophase 1 oda end la. So by the end of telophase there is 2 haploid nucleus that is formed. Now cell undergoes cytokinesis. So cytokinesis For plant cells it is going to be by cell plate method. For animal cells it is going to be by the invagination of cell membrane. So நமக்கு வந்துட்டு நம்ம ஆல்ரெடி அந்த டிவிஷன்ஸ் எப்படின்னு பார்த்துட்டோம். So it is just formation of two haploid cells. Haploid because homologous chromosomes are separated to both the separate nucleus. So இங்கே cells வந்துட்டு they are going to become haploid. So ரெண்டு haploid cells form ஆயிருக்கு. But we have to remember these haploid cells, they have their sister chromatids, that is their replicated copies are present in these cells. So these replicated copies are what is going to be separated by meiosis 2. So now the cell is going to undergo, after cytokinesis 1 there is going to be an interkinesis stage, that is a short preparatory phase. It is going to prepare all enzymes, ATP, RNA, everything required for meiosis 2. So in meiosis 2 the first step is going to be karyokinesis 2. In karyokinesis 2 the first stage is prophase, then is going to be the metaphase, then is going to be anaphase, then is going to be telophase. So all these are representing the second meiosis, and then is going to be the cytokinesis 2. So cytokinesis la there would be two cells formed. So karyokinesis 2 starts with a haploid cell. So cytokinesis 2 is going to form two cells which are also going to be haploid. So this meiosis 2 alone separately without meiosis 1, ஏன்னா meiosis 2ல தான் வந்துட்டு haploid cell திரும்பவும் புது haploid cells கொடுக்க போகுது. whereas meiosis 1ல வந்துட்டு it was in cytokinesis 1 diploid cell, இந்த செல் வந்துட்டு diploid cell, இது ரெண்டு haploid செல்லா மாறி இருக்கு. So this meiosis 1 வந்துட்டு is a reductional division. Actual reductional division வந்துட்டு meiosis 1 is a reductional division, that is 2n cells are converted into n cells, whereas meiosis 2 வந்துட்டு it is haploid cells that are getting converted into haploid cells again. So this can be called as equational division. But on the whole meiosis அப்படின்னு கன்சிடர் பண்ணும்போது starts with 2n cell and ends in n cell, so called as reductional division. நம்ம ஒரு ஆவரேஜ் மாதிரி கால்குலேட் பண்ணிக்கிறோம். ஸோ ஸ்டார்டிங் வந்துட்டு 2n cell, ஆனால் meiosis 2 முடியும் போது n cell, ஸோ reductional division. ஆனால் இதே வந்துட்டு meiosis 1 வந்துட்டு starts with 2n cell, ends in n cell, ஸோ reductional. meiosis 2 வந்துட்டு starts in n cell, ends in n cell, ஸோ equational division. The meiosis 2 in the process வந்துட்டு is similar to mitosis happening. ஸோ நீங்கள் mitosis நான் ஃபஸ்ட்டு சொன்னேன் இல்லையா, the same mitosis process if it is happening with a haploid cell நடந்தால் எப்படி நடக்குமோ அதே மாதிரி தான் meiosis 2 நடக்க போகுது. ஸோ இட் இஸ் ஆக்சுவலி கோயிங் டு பி இன்டர்ஃபேஸ், தட் இஸ் meiosis 2ல வந்துட்டு there is no interface, அதாவது G1 S phase G2 phase கிடையாது, அதுக்கு பதிலாக வந்துட்டு preparatory phase மட்டும். It has already a replicated copy, so it is going to start with an N cell with its replicated copy. So with its replicated copy start ஆக போகுது. So In prophase is normally what happens in prophase, that is the nuclear membrane disintegration, nucleolus disintegration, organelle disintegration இதெல்லாம் நடக்கும். newly formed structures வந்துட்டு spindle apparatus, அப்புறம் மெட்டாஃபேஸ் பிளேட் மெட்டாஃபேஸ் 2ல வந்துட்டு ஈக்குவட்டோரியல் பிளேனில் அரேஞ்ச் ஆகும். அனாஃபேஸ் 2ல வந்துட்டு சிஸ்டர் குரோமாட்டிட்ஸ் செப்பரேஷன் நடக்கும். ஸோ இன் அனாஃபேஸ் 2 தேர் ஆர் சிஸ்டர் குரோமாட்டிட் separation. So there is interzonal fiber formation in anaphase 2. அதுக்கப்புறம் telophase-ல திரும்பவும் reformation of new nucleus. Cytokinesis 2 in plant cells by cell plate method and in animal cells by invagination of cell membrane method. For new 2 cells are formed which are going to be haploid. So this process is meiosis 2. As a result of meiosis, ஒரு single diploid cell வந்துட்டு, so there was only one cell that is taking part in meiosis process, resulted in formation of 4 haploid cells. The importance of meiosis, this is to maintain the species specific chromosome number. Species specific chromosome number அப்படின்னா ஒரு species வந்துட்டு if it is a diploid organism, for example, if the diploid condition of an organism is 23 pairs. இப்போது இந்த 23 pairs வந்துட்டு next coming generationsல maintain ஆகணும் அப்படின்னா it is done if the organism is undergoing sexual reproduction. Sexual reproductionல gametes are formed by meiosis. அப்போ இந்த 2n cell வந்துட்டு என்ன ஆகும்? So it is a normal cell. reproductive cellsல தான் meiosis நடக்கும்னு படிச்சிருக்கிறோம். So இந்த normal cells வந்துட்டு diploid cells வந்துட்டு they are getting converted into gametes which are going to be haploid. இது suppose this is happening in a male organism, the same is going to happen in a female organism to form gamete. So these two gametes are going to fuse to form a zygote. So in the 2n cell திரும்பவும் 2n cell form ஆயிடுச்சு. This is what is called as species specific chromosome number maintenance, that is by gamete formation that happens by meiosis process. இதுதான் meiosis ஓட importance or significance. And types of meiosis is gametic meiosis. This is meiosis and formation of gametes. இது இந்த processல this happens in organisms that have a diplontic life cycle. And zygotic meiosis is meiosis in zygote to form a haploid cell. So diploid zygote வந்துட்டு it undergoes meiosis to form haploid cells, and this happens in organisms with haplontic life cycle. And sporic meiosis, it is for formation of spores, and this happens in organisms with haplodiplontic life cycle. We will see the explanations of diplontic, haplontic and haplodiplontic life cycles when we study the unit plant kingdom. And now we have completed the unit of cell cycle and cell division, and for your further doubts and clarifications please do contact us. Thank you.